So these are the video notes on the 2.5 reading notes. Some of the things that I'm going to cover in this video are some extra notes about the cell cycle, cancer and its treatment, talk to you a little bit about the difference between chromosomes, chromatin, sister chromatids, etc., just to clarify a few things. And then I have a few notes about the model of mitosis that is in your book. Just make sure we understand how it's a model and how there might be some things that are a little different in it than what really happens. Alright, so let's go ahead and get started. So the first thing that I said I would cover was the cell cycle. And so what I hope that you have in your notes is this circle, as best as, I don't have a very steady hand, so I struggle with this a little bit. That should be more like this. Alright, and just adding a few things here. So, just want to add this section that I'm going over right now of the cell cycle, just between this point and this point, and let's say that the cell cycle is going in this direction. So this is G1, this is the S phase, and this is G2. And all of that, the orange, are parts of what we call interphase. And notice that it takes up most of the cycle. So most cells spend most of their life in interphase. And some cells actually, once they're formed, only live in interphase and don't ever go through mitosis, or they stop going through mitosis at some point. For example, nerve cells or brain cells. Once they're formed and have made their connections, the reason why brain damage cannot be repaired at this point, because we don't know enough about stem cells, is simply because it's not going through this phase over here, which is the M phase or mitosis, and then immediately after mitosis is cytokinesis. And in case you don't know, cyto, we've seen a couple times with endocytosis and cytoplasm. Cyto means cell. Kinesis, think of like kinetic energy, it's moving, so the cells are moving or splitting apart. And so these are the phases of what's happening, but I just want to go into a little more detail that in this cell cycle. So, and interphase, because the book goes into some detail about mitosis, and that's what you need to focus on. Let me give you some extra information about G1, S, and G2. So G1, we'll do it this way. So G1 really stands for pre-synthetic gap. Alright? And this is really where the cell grows in size. When the cell starts in G1, it's the smallest it's going to be. And the cell grows in size. It's still doing its normal functions. It's part of interphase. Depending on what kind of cell it's specialized or differentiated into, but it's doing its job in G1. And this is really where the cell grows in size. In S phase, which stands for, which sometimes is called the synthesis phase, is when DNA is getting copied. So DNA replication is happening. Now, one thing that the book will say is that the chromosomes are copied, but we'll talk about this in a second. It's actually not the chromosomes, because the DNA is not in chromosome form yet, but it is DNA replication. DNA is getting copied in DNA synthesis. And in G2, which is called the post-synthetic gap, and that's where the G comes from, organelles are copied, or double. So this is when chloroplast mitochondria replicate on their own because they have their own DNA. So organelles double, and there are some final checks that happen here before the cell divides or starts the division process of mitosis. So that's what's happening in cell cycle. I hope that clarifies a few things in the process. I'm not going to go into the mitosis cytokinesis. This is pretty well covered in your book. You need to make sure that you understand PMAT, or prophase, metaphase, anaphase, telophase, and what's happening inside of the cells, be able to describe what happens in each phase, and be able to identify cells that are in different phases. So if I gave you a cell that's clearly in anaphase, you'd be able to tell me that it's an anaphase and be able to explain to me how you know that based on what you see. Now, I want to actually spend some time focusing in on this S phase, because this connects back to something that we've done before. So I'm going to move on to a new slide here. S phase. This is this is where DNA replication happens, or when, sorry, not where, when DNA replication happens. But in this part of interphase, DNA is not condensed into chromosomes. Now, if it sounds like I am speaking a different language, let me just draw what you might remember as a chromosome from Bio 1. You remember those X's that fit inside the nucleus, or that we see move across the cells? You see all these in the book. DNA does not look like this yet. At this point in the S phase, DNA is just very loose. It's more thread-like. So it's actually sitting in a form that we call chromatin. Now, chromatin, I think of it as like thread, whereas chromosomes are more like yarn. So, in order to make chromosomes, which are condensed, this chromatin has to be wrapped around itself to make the chromosomes. Now, let me show you a diagram that's going to explain this in a little more detail. Alright, so this is a diagram that I pulled offline, and we'll see how this works. Don't know if it will, but what we have here is, I don't know if I can write on top of this. Okay, so I think I figured that out. So what we have here is a diagram that kind of zooms in on our chromosome. So here is that X shape that I was talking about at the bottom. And notice that the width of it is 1400 nanometers. That's 1.4 micrometers. So very, very small. Excuse me. So if we zoom in on a part of this, a very small section, what's here in red, we would see something like this, where this genetic material, or DNA, that makes up this chromosome is wound up and all spirally here, and it spans a width of 700 nanometers. So we're looking at basically the width of one of these arms of the chromosome, and this is the way that it's configured. Now, if we were to zoom in on this, we would see DNA that kind of loops up and down like this, that's 300 nanometers in width, and if we zoomed in on a small section of it, we would see DNA wrapped a couple of times around these things called histone proteins. But they're these histone proteins are associated with each other. Notice that it says that 30 nanometer chromatin fiber is packed nucleosomes. So this DNA with the histone protein is called a nucleosome, but it's just wrapped around each other. And then if we were to kind of zoom in on just these three, let's say, and pull them straight, we would just have this beads on a string. But it's got DNA wrapped around it. We're talking about 11 nanometers here. And then if we zoomed in on just a section between these histone proteins, we would find our double helix that we talked about. Now notice or think about this, if the DNA is all wrapped up and condensed and wrapped around itself to make our chromosome, helicase and DNA polymerase are not going to be able to get in there to do what they need to do. So when DNA is sitting in the S phase of, or any part of interphase, DNA is doesn't look like chromosomes. In fact, it's all loose threads, more open DNA, so that the enzymes that need to come in to do transcription and translation as well as DNA replication can do so. It's only in preparation for mitosis that the DNA starts to wrap around itself to make these chromosomes, to make it a little bit easier to split the copy of DNA in the two separate daughter cells that are made. So when we have our chromosome that looks like this, it actually, the whole thing is referred to as a chromosome, but sometimes it's referred to as a metaphase chromosome, and each one of these sides is called a sister chromatid. And the way that we need to know about sister chromatids is that they are exact copies of DNA, and that they resulted from DNA replication, which should get in your mind helicase and DNA polymerase. They're exact copies unless a mutation occurred. But these are sister chromatids, and the reason that they're called metaphase chromosomes is that after anaphase, they've split apart. And they're still called sister chromatids, but they're also referred to as anaphase chromosomes. So our definitions and our terms change depending on what phase we're talking about, but essentially note that these anaphase chromosomes are genetically identical to each other. It just depends on what phase we're talking about. Okay, so and one last note about the models of mitosis. All right, so I just want to point out that let's see, this is not okay, that the models in your book of mitosis, and I'm not going into that again, that's what you need to know and read, make sure you understand, use 4 chromosomes, and that this is a very simple model, although for some organisms it may be true, but I want you to really think about what this looks like in humans. Because we have a whopping 64 chromosomes. That doesn't mean that we're any more advanced than something that has only 4 chromosomes, but we just have more compartments of genetic material that we have to account for, and that we have 46 of them. So we have quite a bit going on inside those cells for mitosis. So know that this is a very simple model, and that different species have different numbers of chromosomes, or they may. So really how complicated those cells look may be very different. Okay? And I think that wraps us up.